For the next hour, hour and then a break and then another hour, we're going to be talking about the field data. This is our chance to kind of get introduced to the in-situ data that was collected for the Delta X campaign. It's also our chance to work out any issues with Google Colab, Google Drive, etc. So please feel free to stop me if you have questions. So there's only 2 hours allotted, but there are quite a few data sets that we collected and that we're going to cover. So you guys, I'm going to ask you your priority for which data sets you're interested in, and we'll see how many we can get through in 2 hours. Oops. Okay, hold on. Can everybody in the back see that? Okay. All right, so I have designed 6 Python modules that cover wetland vegetation, sediment accretion, discharge, water level, water quality, total suspended solids, and then some grain size analysis. So you can, everything here in bold, there is an associated Python notebook that we can run through. The other data sets, I will kind of introduce them and talk about what we collected, but there's no Python notebook. The data is either, I mean, it's relatively self-explanatory, so you can definitely explore them on your own. A short description of each of these modules are shown in the boxes. And I kind of want to get an idea of if people are more interested in the biomass, the accretion rates. The modules are pretty fast, so if we don't get to them, they're easy to run through on your own. They're well described, and I think that you can all handle going through them if we don't get to them. So does anyone have a strong feeling for a particular data set that they want to see definitely in the 2 hours? Yes. The water quality, okay. The discharge, ADCP, and the gauges, okay. Okay, Rory? The sediment accretion rates, okay. I mean, they're relatively fast, so if you guys feel like we're moving along, we can definitely get through all of them. It'll just be rushed. Yes, Talib? The transport? Okay, so module 6. Anyone really excited about biomass? Andre doesn't count? Okay. So I have a presentation that will go through each of these and describe them. We will start with the biomass, but maybe we'll move more quickly through it. It's, again, I think if you're familiar with biomass data, it'll be pretty self-explanatory. All right, are we ready to get started? All right, so a lot of the data that was collected in the field was collected at these 7 intensive sites. So these are mostly located at CRIMS sites. Is everyone familiar with CRIMS? No one's not? Okay, good, perfect. The only one that was not at a CRIMS site would be the Wax Lake Delta, and I'm sure you're all familiar with Wax Lake Delta. So the CRIMS sites that we sampled range in salinity and sediment availability, and so that's kind of a big focus on why they were chosen and kind of how we're going to frame the questions about the data that I'm presenting. All right, so we're going to start with the vegetation data sets. So these, what was collected was above ground biomass, below ground biomass, and vegetation structure, so stem density and diameter. Okay, so the first one is above ground biomass. This was collected by FIU, led by Edward Castaneda. If you're familiar with biomass data, it's pretty straightforward. They're quadrats that are made with these PVC pipes. They're thrown on the ground, and all of the vegetation within them is clipped. The vegetation is then sorted by species. Then they also looked at the number of stems, but we'll get into that later. And then the vegetation was dried, ground, and processed for carbon, nitrogen analysis, and phosphorus. So it's pretty straightforward. The data for this is biomass, grams per meter squared, or carbon by percent, nitrogen by percent, and phosphorus. The files for this are just one per field campaign. So you download it. Every row is a species within the plot and gives all of those data values. Okay, so this data set was actually recently made public on ORNL DAC. So now you can actually search for it and find it. It's in the final version, and this is the citation for it. Did anybody download this data set? Right? Did anybody not download this data set? Okay. All right, then we have below ground biomass. So this is the counterpart to the above ground. These are taken with cores collected next to those plots. The cores are 11 centimeter diameter, 50 centimeters deep. They're cut into 10 centimeter intervals. Each of those are then washed to remove the soil. And then the roots are separated into live and dead, and large and small. And then they're dried, ground, and run for carbon and nitrogen. Yeah. Oh, sorry. The one is behind the other. Right, sorry about that. Okay, so yeah, this is a picture of Edward collecting a core. I'm sure many of you are familiar with collecting cores in Louisiana. It's a pretty standard procedure. Okay, and again, this data is available on the ORNL. So it's published, you can download, search it and download it. It's also on the Google Drive, so if you have access to that, you can download it from there. Okay, so we're gonna jump into the first module. So if you go back to that GitHub page, click on module 1. I'm going to leave this point. All right. Okay, so each of these modules, can you guys see, is it big enough or do you need it bigger? All right, so each module that I've made for the field data starts with where it can be accessed. So here again is how you would search for it on the ORNL DAC. A data citation, so each of these data sets have a DOI. And then we also list who the people are that collected the data and processed it. In this case, it's Edward and Elena. And then we kind of briefly walk you through the data set. So these are Excel or CSV files, so you can open them in Excel and do a lot of this in Excel. So if you're not, if you prefer that way, that's totally fine. It's a great way to explore the data and see what the data columns look like. So if you read through this, it again introduces the CRIM sites. So I don't know how familiar you are with them, but they are quite a bit different in terms of the vegetation that's there, the sediment that's delivered there. And so this can, and the hydrology as well, so this can impact the biomass. So we're gonna go ahead and try this out. If everyone could click on this first cell. So these gray areas are actual code versus the other stuff is just text. So if you click in the cell, you can either press this play button, or depending on your computer, you can shift enter. And you should probably see a pop-up like this. So this is, you obviously click run anyway. We made these, Google did not. Does the online version look okay? All right. And this, all right, so the check mark means it's done. When it's like grayed out, that means it's thinking. This cell should not take very long to run. So if it is, then you might have another issue. Does everyone have success? Okay. All right, so this next bit tells you if you were running this on your own computer, so not through Google Colab, you would not want to run this cell. This cell is where we connect to our Google Drive. Once you run it, it will ask for 2 permissions. One is to authenticate your Google account, and one is to allow access to your Google Drive. So I'm going to ask everyone to go ahead and run that. And do, allow the permissions as necessary. Okay, so an important thing here is this last line in this cell is going to indicate where this data is stored. So there are 2 important things here. First is this space in my drive needs to be there, or it may not work. The second is if you kind of went through the instructions before the workshop, hopefully you did the step of adding a shortcut to your Google Drive. So basically, Google Drive can only see things that are on my drive. This is a shared folder, so it is not immediately on your drive. So you need to add a shortcut. And to do that, you go back to the Google folder. Okay. At the top here, you'll see this little arrow. You'll click on that, and you want to add shortcut to drive. When you do that, there's another button you have to click. Everybody have success with that? Anyone not have success? Hampton? Okay. Well, I think we should wait here and make sure everyone's caught up, because this is, this is a pretty big step. Yeah. That particular function will work, but when you actually go to look for files, it won't find them. Yeah. Or if you don't have the space, it won't find them. Seriously. Okay. Anyone else have issues? Maybe, Daniel, do you want to just check on Hampton? Okay, so we go back. Okay, so we're back at our notebook. You shouldn't need to change any of this unless you, I don't know, have modified something. But you should not have the ability to modify anything in this shared folder, so you won't mess it up. Okay, so the next cell is to mount your drive. In my case, it's already mounted, so it will say already mounted. Anyone have issues with that? Okay, so now this next cell is where we're actually going to open a file. And if you have any issues with your connection to Google Drive, this is where you will see it. So what it's doing is it is opening the above ground biomass spring 2021 CSV file. The second line is just simply replacing the negative 9,999 with what Python will recognize as not a number. And then this is just printing out the first 5 rows so you can get an idea of what the data looks like. So this is a good chance to kind of familiarize yourself with this data set. You've got the basin, the campaign ID, date collected, lat long, and site ID. Those are pretty common throughout all of the field data. So they should you should see them repeated on all of these data sets. And then we have hydrogeomorphic zone. Yes, Sarush. Okay, so maybe one of the instructors can troubleshoot? And occasionally it will ask you if you're not a robot. So since this is on Google Space, they don't want you to just sit there not actually using it, since you're using up their computation. Anyone online? The step where you add a shortcut to drive is essential. So it's the one you cannot skip. Otherwise, it won't work. Everyone else good? Yeah. Yeah, so the question is if you can change the allocation of resources, and you can pay for more, but otherwise I don't think so, unless, yes, Talib? How to what? Yes, yes, yes. Yeah, so besides buying more, our experience is that it kind of varies. Sometimes it crashes unexpectedly, especially, and if you have multiple of them open at once, that will all take up from your allotted memory. Yes, there's probably a lot more that we don't actually know about it. I think maybe the person who knows the most is Kyle. He's in the back if, okay. Yeah. All right, so again, for people online, if this is just a reminder of how to add the shortcut to your drive. So you find the folder, and if you can't find the folder, please let us know in the chat so we can make sure you find the folder. And then at the top, you'll want to click on this little arrow, and there's an add shortcut to drive. And when you click that, you have to click again, add shortcut. And you can do this multiple times, and it will show up as multiple copies on your drive, which doesn't matter, but mine probably has 10 versions now. Any other questions while we wait? Yes, so the question is if you'll have access to the folder after the workshop, and the answer is yes for a while. Eventually this workshop will, like the recordings will go, I think on Ornell, and there'll probably be a more permanent link, just because once I'm gone, the folder won't be maintained necessarily. So yes, it will be somewhere accessible. And again, all of the data will be publicly available, so.
One question is how to check the unit for those data online.
That's a great question. So all of the information about the files are in the user guide. User guide is on Ornell. So maybe we'll just use this as an example. If you were to, again, this one is a public data set already. It's finalized. So you can search for it. Okay, so we know the exact name of this data set, but you can also search just for delta X, biomass. You'll see the recent collections. You'll also see 2 data sets from pre-delta X, so 2015, 2016. Okay, so we want above ground biomass. This is what the page would look like for this particular data set. You'll see a big download button that'll download all of the biomass data. You'll also see user guide. So within the user guide, you will find a summary, citation, explanation of how data were collected, and then eventually a table that describes each column of the file. And in that should have the units. So in this case, the total dry mass or biomass is grams per square meter, and all of the chemical analysis is milligrams per gram. Everyone set? Except you sure? All right, so I'll scroll back to where we were. We imported this file, and we can see the first 5 rows. So I'm going to continue through the different parameters. So we have hydrogeomorphic zones. So this basically represents elevation relative to the particular tidal range of this area. There's supertidal, intertidal, and subtidal. And if you read the user guide, it'll explain the sampling design and how which different zones each site has, et cetera. Then there's sampling station and quadrat ID. So those are just different forms of replicates. And then we have the species, vegetation class. So that's whether it's alive or dead. So it would be AGN for necromass if it were the dead material. And then we have the total dry mass, total carbon, total nitrogen, and total phosphorus. So this one's pretty straightforward. Okay. So if we scroll down, there's a little bit more information. So again, these are designed so you can run through them on your own afterwards. We tried to put as much information so you would know what's going on, even if I wasn't here. So basically, the first thing we're going to do in this particular data set, every row is a species, which means that multiple rows could represent one quadrat. And for this exercise, we just want to know the total above ground living biomass. So we're going to sum everything that's in the same quadrat that is living. And that's what this line is doing. So it's grouping by site, hydrogeomorphic zone, sampling station, quadrat ID, and vegetation class. So we're going to go ahead and run that. The next line just resets the index, so you have no indexing issues later on. And now we have the total values for both live and dead biomass within each quadrat. For this, we're just going to look at the living, so we're going to filter out only the AGB values. So again, it helps if you've actually looked at the data and you know kind of, well, A, you know the column names are, B, you know what AGB versus AGN are, so reading the user guide is really important. In this case, all this line is doing is filtering for anything, any row where the vegetation class column equals AGB instead of AGN, which is necromass. Anyone have questions so far? All right, so next we're going to find the average and standard deviation of the total dry mass within each site and hydrogeomorphic zone. So again, Edward collected multiple quadrats within each hydrogeomorphic zone, within each site. So it's a very beautiful sampling design, well nested. And these lines will just take the average, the standard deviation, and then count the number of replicates there are. And it will print out the means. So this is a pretty nice way of looking at this. So you can see at this site, CRIMS 294, the intertidal had total dry mass of 164, an average of 164 grams per meter squared. So obviously you can look at that as a table. You can also plot it. So the next step will be to plot that data. Okay. The plotting is interesting in Python. There are like 1,000 things you can do. We have created very simple bar plots, which just plots the average, and then we make error bars using the standard deviation divided by the square root of the number of samples. You're welcome to play around with this code, change the colors, change the labels. You'll probably run into errors, but usually the errors are descriptive and you can figure out why it's not working and you can make changes. So if we run this, we get this plot where each set of colored bars are a different site and the colors are the hydrogeomorphic zone. The Y value is the total dry mass in grams per meter squared. And all of this beautiful plot came from these 4 lines. Okay? Again, there are lots of ways to plot in Python. This particular package is matplotlib. It's pretty standard. There's lots of documentation, and pretty much every error you can get, you can Google, and someone has an answer for you. Any questions? All right, so discussion question. This is not just for clicking buttons. What trends do you notice in biomass between sites and hydrogeomorphic zones? If anyone is familiar with these sites, maybe they have some insight on why one might have more biomass than the other, or why a different hydrogeomorphic zone might have a different biomass. Does anyone have thoughts about this beautiful plot? Any trend they notice? Andre, I see you smiling. What do you think? Yes, this is spring. Yeah, so he noticed that Mike Island has lower biomass than you might expect. So this is all from spring, so it's before the temperatures have warmed and the plants have really exploded. Good. You'll also notice that Mike Island does not have an intertidal site. You can blame Edward for that. Yes, Telly.
Hi, Alex. Online, we have a question.
Yeah.
They would like to for you to clarify. You went to the CRMS sites and collected your own samples?
Yes. Well, I did not personally go, but Edward and Elena and one or two others went and physically collected these samples, yes. So these are all, I mean, they're all part of the DeltaX campaign. The field data is both a data set on its own, but it's also going to be used to calibrate, in this case, Avarice data and other airborne data, and also maybe help with roughness in the hydrodynamic models. So there are multiple purposes for these data sets. But yes, they were actually collected by, not me, but us. Yeah. Yep. Yes, exactly. So Kathleen pointed out that the map is at the top of the notebook. So you can see the sites. So if you're not familiar with them, you can see which ones are saline. You can also notice, I mean, as Mark mentioned, the big part of this is testing the difference between the basins, so the Atchafalaya versus the Terrebonne. I'm sure you're aware that the Atchafalaya has a much larger sediment load. It's a growing area versus Terrebonne has a lot of erosion and subsidence. So we're also looking at differences there. Obviously, there's a big difference in the plant species that are there between the two basins, fresh versus saline. So there are a lot of things you can explore with these data sets. Okay, so the next thing is the chemical analysis. So we can do the same exact thing we did with biomass, just with the carbon, nitrogen, and phosphorus. This cell just filters by, so not all of the samples were run for chemical analysis, just the samples with A. So we're just filtering out the ones that don't have values. And then we can do the same thing, means, standard deviation, and we can look at a table of those values. So we can see differences in carbon, nitrogen, and phosphorus for each of these sites and hydrogeomorphic zones. And then we can plot this. Okay, so we don't really have time to talk about trends here, but this is another plot that's fairly easy to make. And I'm sure you could spend a lot of time talking about these differences, and I'm sure Edward will be spending a lot of time talking about these differences. Okay, so we're going to move on to below ground biomass unless there are some questions. Okay, so this next one is going to pull the below ground biomass file. Again, if you have any issues with connecting to your Google Drive, this won't work. And we're going to filter for the ones for the live root biomass and exclude the dead root biomass. Okay, so the below ground biomass is a little different. Each core, they have segmented into intervals. So each row in this file represents a different interval between 0 and 50 centimeters. So we're going to take an example of from one core and plot a vertical profile. So this is just filtering for site 396, intertidal live root biomass. And then we're going to group them by the depth interval. So all the cores taken at that site in that hydrogeomorphic zone, we're going to average the values for each interval of the cores. And then we can plot that. So you'll notice this plot is probably sideways from what you would normally see for a depth profile, and that's just simply because rotating it would take a lot more lines, and we want to make it as simple as possible. So the depth interval is on the x-axis, so we have 0 to 10 centimeters up to 40 to 50 centimeters, and the live root biomass in grams per meter squared is on the y-axis. So these two sites, CRIMS 421 and 396, you can see they have quite a bit of different biomass amounts, and you can also look at how that changes as you go down, vertically down the core. Okay, so it's 9:45. All right, how are people feeling about using the Google Colab? Good. Okay, then we're just going to click through these so we can get into the data sets people want to see. We're going to create some more plots. And then finally, that last one just calculates the root to shoot ratio and plots it. Okay, so when you get these, you can actually at the top click Run All, and it will just run everything, and you'll see all the beautiful plots. And then you can kind of work through what you're interested in seeing. So we are going to move on to another set. All right, so in addition to biomass, Edwards group also measured vegetation structure. So within those quadrats, they then split it into quarters and measured the stem density, stem diameter, and stem height of that. So the sampling, oh gosh, sorry. Can you see it online? Good, okay. All right, so stem density and stem diameter for this data set. This one is not yet publicly available, but you can find a link to the preprint on the Google Drive or the data access file. Okay, we're going to move on to sediment. So this was collected by two different teams. So this is LSU and FIU. So the first are soil properties. So FIU again collected cores, slightly different shaped cores, and they measured the soil bulk density, organic matter content, carbon density, total nitrogen, and total phosphorus. So this is another pretty straightforward data set if you're familiar with soil science in coastal Louisiana. This one again is not yet publicly available, but the preprint is available to you all on the Google Drive. Okay, sediment accretion. I believe a few people were interested in hearing about this one. So these are feldspar marker horizons. Is anybody not familiar with a marker horizon? Okay, yeah, so basically you put down the feldspar and then you come back over time and you look at how much material has accreted on top of that horizon. It works great for depositional environments. Obviously, erosional environments, the feldspar can often wash away, which I think happened in a few cases. But in general, it works really well for these sites. So similar design, these were all done at the 7 intensive sites. You can read all about the processing and methods in the user guide. So this was done by the LSU team. It is also not yet publicly available, but the preprint is available on Google Drive, and this is the current citation. Okay, so we are going to move on to the second module. So if you go back to that GitHub page, you will find Module 2, Accretion. Everyone get there? All right, so this is set up very similarly, right, where it introduces the access and the citations and the authors. And again, you can run through these. The first several are the same. You will have to allow Google access again for every notebook and every time you log into Google. There is probably a way to change that in your settings. But for now, you will have to click allow again and again and again. And again, this is, oops.
Could be incentive for installing your own Python Jupyter notebook down the line.
Yes.
And your own data.
That's true. If you run this on your own computer, you don't have to go through those steps over and over again. So here again is the path to the data. So don't change that and make sure that the folder, there's a shortcut to the folder on your drive and that you have this space. Okay, we'll mount the drive again. A reminder of the intensive sites. And again, these kind of all start the same. You upload the file, you kind of take a look at it, you remove any values either that were bad or not collected. In this case, the LSU team uses not collected to represent no data values. So we want to convert those so we don't have any issues with strings floating around in our data frames. So we print out the first 5 rows again, and you'll notice that it's a similar format. So basin, campaign, site ID, hydrogeomorphic zone, lat long. In this case, we also have elevation. And then the data variables. So time marker was deployed is when the feldspar was laid down. Time marker was sampled is when they went and actually collected the feldspar. So that's done with the liquid nitrogen cryocoring, so you can see the horizon in the layer. I think what you'll probably actually want to look at, though, is the normalized accretion. So this is millimeters per year of sediment accretion on top of that horizon. The LSU team then also collected cores next to the feldspar to the depth that was accreted, and looked at soil bulk density, organic matter content, and carbon, and soil, organic carbon. So you can actually see the properties of the material that was deposited in these environments. So this data set was just recently updated, and it now includes 2 years of data. So we're only going to be looking, in this module, we're only going to be looking at the first year. So from 2019 to 2020, the accretion rate. So that's what this line is doing. It is filtering for only those from the first year of sampling. These lines should look familiar. We're again taking the mean and standard deviation of the accretion rates for each site and hydrogeomorphic zone, and then looking at a table of that. So again, you'll see the site, the hydrogeomorphic zone, and the variables that we're interested in. So here is normalized accretion, again in millimeters per year. And you can see the range between the different sites. If you want to plot that, which is much easier to look at, this cell will plot all of those variables. So we have bulk density, elevation, organic matter content, and accretion rate. And you can look at them and draw conclusions. Since this was one of, who was the person interested in the sediment accretion? Okay, is there a value you're expecting, or is there anything, any trends you notice between in this plot? So these are the sites here and the accretion rate. Can anyone throw out a number of an accretion rate they'd expect for coastal Louisiana? Like you're thinking about it, Greg? Okay. Yeah, so these are pretty big, right? They're greater than expected, especially in Wax Lake Delta. But again, this is a delta that's growing, so maybe that's a good thing. Any other thoughts on this data set, on this plot? Andre, do you have any? That's a great question. My guess is most people here aren't like as familiar as you are with 421. Maybe you want to answer your own question? Yeah. Yeah. So to summarize that for those online, next time we can maybe give him a mic, but yeah, so it's possible that the greater accretion rates at 421 are due to some kind of storm deposition. And maybe this has actually been studied by others. But yeah, that's one hypothesis for why this site might see greater accretion rates. Yeah.
Those are the mark horizon data, right? And from long term, to me, that's short term
Yes.
accretion rate. Over long term, say decadal scale, we should use other geo technology like cesium, lead-210
Yes.
data. So to me, those even this wax legs delta, so it's high, it's still reasonable. I mean, after you convert to long-term rate,
Yeah.
may not be,
Right.
like we expected that much.
Yes. Yeah, so obviously this is definitely short-term accretion. And in fact, you can use this to break up a year into month periods and actually look at seasonal effects or storm effects. So yeah, it is not really meant to be a long-term thing.
Right, we collected that data to use in the normal model.
Yeah.
It's a cohort model that builds soil columns. So you start the model with the inorganic sediment load.
Yeah.
And then the model has other parameters that will work that layer through time, decomposition and compaction and all those things. And then you get to the output of the model should be the long-term rates like he
Right.
just mentioned right there.
Yeah, and then I assume you're using what you get from that model to compare with what other people have found from cesium's stuff. I know that the cesium was supposed to be part of Delta X, but it was cut out from.
Yeah,
COVID.
but as part of Amanda's not here today, but she's a master's student with Robert, and what she's doing, we collected cores for dating with cesium, and they're actually being run right now at LSU with Kancha Mighty. And the idea is that we're going to use those cores and other published values that have produced long-term accretion using those cesium and lead-210 in coastal Louisiana to compare with what her model outputs will be.
Right. Perfect. All right, so Oh, sorry, yeah, go ahead.
With this data set with the soil bulk density and organic matter, is that from cores at each marker horizon plot, or is it of the sediment on top of the marker horizon?
Sure, yeah, you can answer, yeah.
So there's two ways you can do that. Maybe there's even more than two ways. One way is you core like right on top of your sediment, your marker horizon, right? And then you take your like a height or depth measurement of that accretion, and then you can slice that right above the marker and bring it to the lab, and then you run all the nitrogen, carbon, phosphorus, whatever you want. We didn't do that. We collected using liquid nitrogen. So you're familiar with that technique? So you put a copper straw, like a hollow copper straw, about, like centimeter, half a centimeter diameter, and then with another hollow copper straw. So you put that on top of the marker, and then you dispense liquid nitrogen pressurized into that hollow copper straw. So the soil around that copper straw freezes. So when you pull it out, you see it looks like a popsicle. So we call it like a mud popsicle, right? You can measure your accretion right there, but the amount of soil that froze around it, you can't really bring that back to measure for bulk density because it's not an intact soil sample, right? So we go right next to the marker, like right around it, like a few meters, like 2-3 meters, and then we collect with a Russian peat core. We collect soil samples that match the heights that we've measured on top of the marker horizon. And the reason why we do those two separate things is to extend the marker life. Because every time you use a Russian peat core that's like 5-6 centimeter diameter plus the blade, when you pull that up, especially with roots that come with it, you would destroy your 50 by 50 centimeter marker like really quick. So that's why the mud popsicle makes less of a damage. And it also gives you more precise millimeter readings. They should be an average because every time we pull a popsicle, we make 3 readings around, and then we have 2 popsicles per marker, and we have 3 markers per station. So that's an average from 6 cores that we collected to measure those 6 averages of accretion that we pulled out. So 2 and 2, 6, right? So each one of those has like 3 measures around. Based on that, we collected the 6 cores, and then that's the bulk density that you get. Is that right? How we have it in the data set? We have an average
Yes.
for each station, right? We don't have an average for each marker inside each
You have
station.
X, Y, Z.
Oh, so we do. So we do have an average for X, an average for X, Y, and Z, which are the 3 plots that make a feldspar station.
But yeah, so everything above the horizon is considered one sample. It's not split then into.
Yeah, no, yeah, it's whatever. So if you go to one site and you have like 2 centimeter accretion and the other one you have 5, that didn't get separated in layers. That was just like whatever accreted over time. Yeah.
Yeah, so it's the average bulk density of the material that has accreted on top of the horizon.
Were there 3 samples per, so there's multiple, like in space samples per or feldspar place? You didn't just take 1 popsicle, you took 3 popsicles per measure,
We call station and plots. So one station has 3 markers. We call X, Y, and Z. So those are like 50 by 50 centimeter squares. So X, Y, and Z. Every time we go to measure, we take 2 popsicles, 2 measurements out of like X and 2 out of Y and 2 out of Z. But each popsicle, because the soil surface is irregular, and you can see that in the soil frozen around the popsicle, right, around the copper pipe, we take 3 measurements with a caliper.
Okay.
So you're going to get like, let's say 1.2, 1.7, 1.3, whatever. And then you average that, and that average value across the 2 popsicles on each X, Y, and Z, that's what will determine how much soil you're going to collect on the surrounding.
That you answered my question. I was just
Yeah.
wondering
Is
how
that-
you accounted for the variability within a plot, because I'm sure
Yeah.
it's irregular. So, have you measured, like, see how your results compare to like a sediment elevation table, which is obviously takes even more measurements
No,
per plot?
we didn't, but we didn't include that for the Delta X part of things. And also, I mean, this project, we're not looking into that kind of information yet, so.
Okay, thank you very much.
Yeah, so all of these data are pretty fresh. I don't think there's been a lot of analysis, especially in terms of comparing with other data sets yet. Right.
We were in the field, like, collecting the 24-month mark from the time that we deployed the markers, like a month ago.
So the rest of this notebook just basically makes the maybe incorrect assumption that these could represent some kind of long-term accretion, and then compares them with the expected sea level rise. So you guys are welcome to run through those last few cells, but I think we're going to take a break. We're obviously not going to have enough time to go through all of them, so you can walk through the description of each field data set and then do the modules if they interest you. But we're going to spend the next 37 minutes going through water quality, and then hopefully ADCP, because I think there was a request for hydrology. Does that sound good? Can you see the right version? Okay. Okay, so the water quality team were mostly from Boston University. They collected many things in many places. So this map shows all of their water quality sites, and at every site they collected water quality indicators. So from a probe, they collected TSS samples, they did surface reflectance, and sometimes they also did particle size. So this first one are the water quality indicators. So this probe goes into the water and measures salinity, turbidity, temperature, and chlorophyll A fluorescence. It sits in the water for 2 minutes, and then they average that to get a value for that site. And the sampling design was kind of just collect like in a lot of places and try to overlap with the airborne campaigns as well. So you'll notice when you open the files, it's not like they collected every site at this time on these days. You kind of have to work through it and look for what it is that you want to use. So I believe this data set is still in preprint form, so you can get it on the Google Drive or through the links that we sent you, and then it will become available shortly. So then at the same time, they collected total suspended solids. So this is done by dropping this into the water to a certain depth. I think it was about 50 centimeters. And then you kind of snap the line and the ends close, so you collect a sample at that particular depth. Those samples are then filtered to get the total amount of suspended particles or solids within that water sample. Depending on where they were, there's obviously higher or lower sediment concentrations, so they take different volumes of water in order to make sure they have enough sample to process. Samples are dried and weighed, and that gives you the mass per volume of water. So the TSS data is also still in preprint form, so you get it through the link or the Google Drive. And this is the citation. Okay, so water surface reflectance. So those first two, the water quality indicators and the TSS, are what will be in the module. The next two are not in the modules, but they will link to what Daniel is going to talk about this afternoon. So this is just kind of a summary of what was collected, but we're not going to go through this data set. So they use a portable spectroradiometer to measure the reflectance. They use different references in order to calibrate the water surface reflectance. And this was done at every site that they collected those water samples. So all of these four that I'm going to talk about are all coincident. So the sample IDs are how you will link them all together if you want to use the data sets in conjunction. Again, this data set is still in preprint form, and you can find it on the Google Drive or through the links that we sent. And then finally, the beam attenuation and particle size. So this is done using a list. This instrument here, it's put into the water, I think all of the same depth, and they can measure in situ the particle size of the suspended sediment, suspended solids. Again, put in for 2 minutes and then averaged for those 2 minutes. And again, collected at all of the same sites. So this is a pretty great data set because they have instruments, they have actual water samples, and they're all kind of done by the same team at the same time, so they work really great together. Okay, and then this data set is also in preprint form. You can find it on the Google Drive or through the links that we sent you. Okay, so we're going to jump to module number 5. Does it look good on the online? Perfect. Okay. All right, so again, the top part of this notebook will introduce the data set where you can get the data and the citations and the team that collected everything. Okay, so go ahead and run this first few cells again to get ourselves connected to Google Drive. So you'll notice at the top of this one, there's something called raster IO, which is imported in a slightly different way. And this is for Collab specifically because it does not come with this package. And so you'll probably see these extra lines of code, and that's fine. It's basically just downloading this package so that you can use it on Google Collab. And in the future, you don't need to do that every time. But sometimes it's easier just to leave it in there so that you don't run into any errors later on. Okay, is everyone up to this point? Okay. So the first thing we're going to do is import a base map. This base map is just an older version of the digital elevation model. So it's just so that we can kind of see what the landscape looks like when we map some of these samples. And this is where we'll use that package, raster IO. Okay, so we're going to be looking at the water quality indicators, which were collected with the probe. And we're also going to be looking at the total suspended solids, which were collected from an actual water sample. So the next two cells basically import those two files. Again, if you have had issues connecting to Google Drive, this is where you will find errors. You'll notice, again, we're replacing this value not collected with an actual NaN value that Python can understand. And then you'll notice sometimes when you import these CSV files that not everything will come in as a number. It'll see things as strings, and that can cause errors when it tries to do math with things that it thinks are just letters. So this line here is basically just ensuring that it knows that these columns are numbers and not strings. Okay, so as I mentioned, these two data sets were collected simultaneously, so their sample IDs should match. And we can basically take the two data sets and merge them together, matching them by their site IDs. And that is what this line does. And then it also prints out, again, the first 5 lines. So we can see what there is. So just as before, we've got basin, site ID, campaign, lat long, date, time. The depth is the depth the sample was collected. And then we have, for anything that was collected with the probe, there is already standard deviation, and that's based off of that 2-minute collection period. So we have turbidity and its standard deviation, salinity, temperature, chlorophyll A, and then if we scroll all the way over, we should see the TSS concentration, and the TSS is the one that's from the water sample. So turbidity is basically, that comes from the probe, and it's a number that is not actually connected to a physical measurement, right? So it's a digital measurement, but it more or less represents TSS in a lot of ways. So those two should be similar, and later on we will kind of compare them to see if we can create a relationship between the two. But you usually use a TSS sample, water sample, to calibrate your turbidity instrument so that when you measure turbidity, you actually know what it means physically. Okay, this is just a little note because I had some issues when opening some of these data sets that you want to make sure you know the names of your columns. I think Ornell has fixed this, but there were a few issues where there were spaces in the column names, or there weren't spaces when there should be. And so if you run this line, it will print out all of the columns that are in this file exactly so that you can make sure. Obviously, Python is very specific. So if you call the column with temperature and you have a space, or you spell temperature wrong, it won't work. And I think that's some of the most common errors or small things like that. So this is a good way to make sure you know what data is there. Okay, everyone good? Okay, so the first thing we're going to do is we're going to take the average of turbidity, temperature, salinity, chlorophyll A, and TSS for each of those sites. And we do that with this line. We group them by site ID, and then we take the average. And then we're going to map that. So this bit of code here basically plots our base map, and then on top of that, we are putting our water quality data points. This will take a little bit longer to load just because of the map, but okay. So we have two. On the left is going to be turbidity, and on the right is going to be salinity. So what trends do we notice? The color bars here, so the darker colors in turbidity are higher turbidity and higher salinity. So there's some pretty obvious trends, especially between basins. Does anyone see anything obvious they want to mention? No? Okay, so on turbidity, we see much higher or redder values in the Atchafalaya. This is expected, right? It's a sediment-rich environment. Lots of sediment coming through the Atchafalaya River and the Wax Lake outlet, as compared to Terrebonne, where we see much lower turbidity values. In terms of salinity, obviously in the Atchafalaya, we have an entirely fresh system, so we have very low salinity values. And then in Terrebonne, we have much higher salinity, as expected. So the closer you are to the coast, the higher the salinity. Okay, so now we can compare those quantitatively. So we're going to take the average of all of those parameters by basin, and then we're going to plot that. So these plots just compare Atchafalaya to Terrebonne. Again, we see higher turbidity in the Atchafalaya compared to the Terrebonne, higher salinity in the Terrebonne compared to the Atchafalaya. You can also look at temperature. scroll down, chlorophyll A fluorescence, and total suspended solids. So again, the total suspended solids trends should match that of turbidity since they represent the same phenomenon. Okay, so then I just have a small example here where we're taking a particular site and we're going to look a little bit closer. So CRIMS 421 is the one that I have chosen. So these lines are just selecting all of the rows from CRIMS 421. And then I have, on my own, kind of decided that 2 of them occur in the large channel and 2 in a small channel. And they also are sampled at different parts of the tide. And I'm going to plot these for you so you can see where they are. Okay, so this is part of CRIMS 421. These 2 samples fall in this larger channel. These 2 samples fall in a smaller channel that isn't really resolved in this map. And we might expect differences in these parameters also between rising tide versus high tide. So just as an example, we can plot those samples next to each other. So I've called them large and high tide, small and rising tide, small, high tide, etc. Yeah, I mean, you can read. So this is just an example of how you can look at a particular site and compare these parameters at different times in the tidal cycle, or maybe what's flowing through the big channel versus what's flowing through the small channel. It's just an example of different ways to plot these data. Any questions? Okay, so the next thing we want to do is see if we can build a relationship between turbidity and TSS. So again, TSS is often used to calibrate your turbidity instrument because turbidity alone doesn't necessarily give you a mass per volume that you might want. So we are going to plot all of the data points, turbidity versus TSS, and see if we can find a relationship. So we're running a linear regression. The dots are the data points. The blue line is the fitted line. And the black line is the 1-to-1 ratio. So basically what you can do, if you then take your turbidity meter out in the field again, you can use the equation for this blue line to convert your turbidity values to actual total suspended solids. Does that make sense? And then we can also look at the other parameters and compare them. So would we expect turbidity to increase with salinity, for example, or would we expect chlorophyll A to increase with turbidity? We can compare all of those. I have picked 2 for this example. So that's what these 2 plots are. So they're all of the water quality points. The first plot on the left is chlorophyll A versus turbidity. So it's nothing like wowing, right? We see a slight trend where higher chlorophyll A means lower turbidity. And maybe higher chlorophyll A is also associated with higher temperature. And these aren't like causation relationships necessarily, but it's kind of a starting point for looking at these data and trying to draw spatial conclusions and how these parameters interact with each other. So again, if you take this cell and you just change the parameter here, you can plot different ones and see how they look. So maybe we want to look at chlorophyll A versus salinity. So now the plot on the left is salinity versus chlorophyll A. So we kind of see an increase in chlorophyll A occurs at lower salinities. So that's kind of the types of things you can do with these data. Obviously, there's a lot of other things you can do. People have any questions or things they want to try? You're welcome. I guess we didn't talk about this, but if you want to make changes to these notebooks, you can. on Google Colab. If you want to save them, you need to save it to your own computer or a Google Drive. Like you won't ever overwrite what we have put here, but you're welcome to go in and make changes, see what happens, change your plots. So maybe we'll take a few minutes to do that, play around with it. Is anyone having any issues? Yeah. Yeah, so if you save it to the drive, you can then basically go to your drive and double click and it'll open back up in Colab. If you're more familiar with Jupyter Notebook or Python on your own computer, you can download the file and it will run. It's the same format as a Jupyter Notebook, so you can run it there as well. But if you want to be able to just kind of play with it online, the drive is probably the easiest place.
Yeah, I saw that it was fluorescence. Do you have the concentration measurement based on the fluorescence? Do you need to calibrate the data?
So I don't know if they collected chlorophyll A. They're obviously not here. I'm not sure if they collected actual samples. I would have thought that they did, but it's not currently available. So. Okay, who was the one who wanted to talk about hydrology? Were you talking about the ADCP or the water level gauges? Okay. Well, is there one you have a priority for? Because we've got 15 minutes. Okay, yeah. Yes.
I'm just interested if you can talk to the temperature disparities between the small and the large, because the samples look like they're all taken on the same day.
Again, this is not a data set that I personally collected, but my guess is a larger channel will they're often deeper. So shallower channels are going to be more impacted by the solar radiation and warming up. They're also not flowing as fast, but are you talking about at that particular site or just the whole campaign?
Even within that small channel, there's like between the rising tide and the high tide. I'm just interested in, that there's quite a large difference, like 4 degrees Celsius.
Yeah, that's definitely true. I can't personally say why that would be, but okay. Hold on, Jan, can you bring the mic to?
Sorry, we're asking everyone to speak to the mic so the people online can hear your questions.
I want to ask a question about, can you suggest a place or way to find out the information on how the turbidity data are derived? For example, the algorithm to derive turbidity from the reflectance data. If that's the way it was derived.
So the turbidity is just measured with a turbidity meter. So it's whatever the instrument itself does. You can find out the specs on that. If you're talking about then the actual reflectance measurements from the water and how we convert that to turbidity.
Yeah, I think I see a map somewhere from the collab notebook that you have a map for turbidity. Or maybe I missed.
The one I showed, the one in this module?
Okay, maybe not. So I thought it was turbidity, but it turns out just a map. So
Yeah. So the map in this module, the dots are the turbidity. Everything in the background is just elevation, kind of.
Hey, Alex.
But yeah, I think.
Oops, sorry,
Yes.
maybe loud. I just want to refer people to the data management plan where all the details can be found. Right?
Yes. Yeah. So each one of these also has a user guide that will explain how all of it was derived. But yeah, in this case, the turbidity is just what's read by the probe. There's no additional algorithm that we use. Okay, so we're going to quickly go through the ADCP data. All right, so the ADCP data is used to measure discharge in channels. This is finally a data set that I did help collect, so maybe I can answer more questions. For now, we're just going to be looking at the spring data. This was collected by Bill Gibson at LSU and by the Caltech team. So basically, you run the boat with the instrument attached either on the boat or next to the boat across the channel very slowly, and you're measuring velocity and depth of that transect, which can then be converted to discharge. Usually we do 2 replicates and try to average them as long as time allows. Occasionally we're trying to collect as many as we can while the airplanes are overhead, so we don't always have replicates. And we use 2 different instruments, and the data is processed in either Wind River 2 or River Surveyor Live. And the output from that is what is more or less what is available for download from ORNL. For the spring data sets, we had some issues with GPS. So all that's available is the discharge and the river depth. The fall campaign will have much more information about the actual velocity profiles and the river bathymetry. So we've converted it to an actual elevation. So this is probably slightly more complicated for downloading the data. So there's one summary file that gives you information about each transect. So the time, location, the total discharge. It also breaks it up in between to the top discharge, the bottom of the channel discharge, and the sides. And then each transect will have its own individual file. And those files contain the location, for each location along the transect, a river depth. And in the future, the fall data will also include each point along the transect will be a velocity profile. So you can basically see every cell, every vertical cell, the velocity in that cell. So if you really want to dive deep into like flow paths along this transect, and like the flow is mostly at the top or the bottom, like that's something you can dive into, but we're not going to talk about that today. So this data set spring was temporarily on ORNL publicly, but it is in the process of being combined with the fall. So now it's only in preprint version. So hopefully you can access that on the Google Drive. And this is the citation. Between the spring and the fall, there were 4 teams working on this. So it was quite a large effort. Okay, so we're going to quickly jump into the ADCP module. You can find it on the GitHub. Okay, again, the first few cells are just importing packages. There's also a nice diagram here of what we mean when we're taking a transect and how the ADCP is working. So if you're not familiar with ADCP, this is a good way to kind of visualize what's going on. All right, so again, we import our packages. We connect to our Google Drive. There is the path where these data are stored. Okay, so the first thing we're going to do is, again, import that base map that we'll use for making some pretty pictures. And then we're going to import the summary ADCP file from the Spring. So again, this line is just to make sure that everything is seen as a number, not a string. And you'll see this top, the first 5 rows. It's a bit hard when we're zoomed in like this, but again, basin, campaign. The file name is basically, if you look at the summary and you're like, I want more information about this transect, you take the file name and you go into the database and you can download the individual file for that particular transect. The transects are also, they're given a general location, so kind of which basin or if it's an intercoastal waterway, and then they're given a site name. Also have replicate. And then you can get lots of more information about the actual setup. So boat speed, the mean speed of the water, and then here is the discharge broken into pieces, like I mentioned, top, bottom, side, and total. And then the average flow direction. And then there's also information about how this was collected. So obviously, each team had a slightly different setup of where the ADCP was mounted, and then also which instrument they were using, which type. And again, the Spring ones had some issues with GPS, so you'll see it in this note section, just letting you know that the exact locations are not available. Okay, so we're going to take, just to get an idea of where these transects are, we're going to take a mean of all of them by site name. So again, this groups them and then takes an average discharge. And we're going to plot them on our base map. Depending on your computer, sometimes Collab is faster than your computer, or if you have a really nice computer, Collab is slower. Okay, so these are all of the transects collected in Spring 2021. They are represented as dots in this case, so that's one of the endpoints of the transect. The color indicates the average discharge. So again, each site was measured multiple times. So if flow changed direction, this will average out to be low, but this is mostly just to visualize. Kind of hard to see when we're zoomed in, but you'll notice that the greatest discharges are through the Wax Lake outlet, which is what we would expect. And they decrease as you go kind of into the Wax Lake Delta. And then we have very low discharges out in Terrebonne. Obviously, this is expected because it's not dominated by the river flow. So the discharges are driven primarily by the tides. Okay, so there's a lot of things you can do with ADCP data. We just have an example of how you could maybe use it for connectivity. So we're going to look at the Wax Lake outlet. We're going to look at discharge kind of at the top, and then in the middle, and then down at the delta. And if that discharge changes, what would that mean? Anyone? So if at the upstream end of the Wax Lake outlet, you have higher discharge than you do at the lower, where has that discharge, where has that flow gone? Right, so it's either gone into smaller channels or it's overflown into the wetlands. So this line is we're going to just take out a few of those transects along the wax lake outlet. And we're going to compare the discharges. So, okay, yeah, so these occur along the wax lake outlet. I'm going to scroll down. This just talks about the tide. So if you want more information about the tide gauges, so you can tell if these transects were taken during high tide, low tide, you can find them here. Okay, and then we are going to further subset those wax lake outlet transects to a specific time. And I wouldn't worry about this code, but I'm basically pulling out a specific day so that we know that the tide isn't changing and the discharge are relatively close together. They're measured relatively close together in time. So since these transects were taken around the same time, differences in these values could indicate fluxes in and out of the wax lake outlet. And we can plot that on a map. I'm going to zoom out just for now, but these are those 4 transects. You'll see there's higher discharge at the top near the intercoastal and lower discharge as you get to the delta. And wax WLOE occurs after this first distributary channel of the wax lake delta. Okay, so that's, I mean, you can speculate about why that might be. Maybe flow is going into these little channels, etc. Obviously, there's not too much flow going along the intercoastal because these 2 values are fairly similar. Okay, the next example is kind of similar, so I'm going to go through it quickly. But basically, we do the same thing back at the CRIM site 421. Okay, so these are a few transects that are collected at that CRIM site. And as you see, they kind of occur along different parts of the channel. So again, we're going to average for each of those transects, and you'll see there's quite, well, there's quite a bit of variability. Yes, Kelly.
There's a question online, if you know how deep is the channel? The depth of the channel.
I don't know off the top of my head, but someone here.
So what is the temporal resolution of this data? Like the transects, is it's taken once or just season basis? You just take data on those transects?
So we return to each transect multiple times. It's mostly determined by when the airplanes are flying or just when the field team can get there, like physically moving between transects. Each time we visit one, we take 2 replicates. So it's kind of, it's not random, but we are trying to capture different periods of the tidal cycle. But it really just depends. I mean, you have to look and see what each one is kind of different.
And how long those data was been taken, like how long, like is it for 1 year, 2 year, or?
They were taken during the 2 field campaigns.
Okay.
Yeah. So for a 1 to 2 week period in the spring and then the fall. And in the spring, there was really only 1 team, so there are less samples.
Thank you.
Excuse me, is there any correlation with the total discharge rate that recorded by the USGS daily water discharge data and the data we measured here? Is there any correlation, like, did you look into that?
That's a great thing that you could look into. Again, this is only data that's been just recently cleaned and put online. So there hasn't been a lot of in-depth analysis, especially for answering research questions or comparing with other data sets. That's something that can be done in the future, but at this point, we're just kind of presenting the data itself.
Hey, I also have a quick question. Did you say earlier that we would have access to the velocity profiles based on depth?
Yeah, so the spring data set, which is all I have here, does not have, well, it has those, but there are issues with the GPS that make it basically unreliable, the velocity profiles. I mean, they can be used to make a total discharge, but their exact location isn't reliable, so we haven't made it public.
Okay.
In the fall, which I believe you should now have access to the fall data, I think it just went online on Friday, that we do have the velocity profiles, and you'll have to kind of read about how the file is formatted. It's a little bit confusing, but basically, yes, you can get, at every point along a transect, you can get the vertical depth, and then each cell, so we call them cells, right? Every interval of vertical depth, you can have the velocity.
And
So speed and direction.
kind of getting onto the previous question, that was done like twice in a day, right? But each transect was maybe replicated once, like that day.
Again, it varies.
Okay.
Sometimes we got to the transect 3 times a day. If we were kind of in a smaller area, we could repeat multiple times. But yes, once a day is probably more likely.
Okay, thank you.
Okay, so due to time, we don't need to go through all of these, but basically we, again, can look at connectivity by comparing discharge values in one channel. And we can get an idea of how much water is leaving the channel to go into the wetlands or going from the wetlands into the channel. So you're welcome to run through those examples. I'm going to skip to the bottom so we can look at some cross-sectional shapes. So again, in the spring, because of the GPS issues, we don't have an absolute bathymetry or elevation of the channel bed. We only have depth, so relative to the instrument. But in the fall, you will be able to get an actual estimate of elevation or bathymetry of those channels. So we're going to open 2 individual files. So I've picked 2 transects, one in the Wax Lake outlet and one in the Wax Lake delta. We're going to look at the difference in their channel morphology. So if you run, I don't know what number this is, but it's after this for the last portion of this module. So if you run this, you can get an idea of what these individual files look like. So the ones that end in bathy mean that they are depths. The ones that end in velocity, which again will only be available for the fall data sets, those are the velocity profiles. And they're all stored in individual files just because they're already fairly complex. Okay, so we can plot these 2 transects. This has 4 plots, so it might take a little bit of time, but here we go. Okay, so on the left, top left, we have the channel shape. So X is distance traveled. Again, you'll notice this is all relative, right? So it's in meters, but it's not, there's no GPS location for these. And then the y-axis is missing a label, but it is the depth in meters. So Wax Lake outlet is 20 meters deep at this point. And it's a fairly simple channel shape. On the right is just showing you where that transect is located. If we scroll down, we can look at an example of a transect in the Wax Lake Delta. So this is fairly high up in the delta, but it is within one of the distributary channels. And you'll notice the channel shape is quite a bit different. And the Thalweg is definitely on one side, not in the center, and it shallows out towards Mike Island. And then this is the location of the transect. So this is just an introduction. You can try pulling in different transect files, especially if you compare channels in Terrebonne. They're quite a bit different than what you find in Atchafalaya. But due to time, I think we're going to finish up here. The slides are online, so you're welcome to scroll through all the other data sets. And if you have any questions about those, you can ask me during a break.